Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today we are going to see how HCl is going to be produced in our intestine or stomach, we can call it. Uh, this HCl is also called as gastric acid. So this is HCl is the best known component of our gastric juice, and either you can call it HCl or hydrochloric acid or gastric acid, it's same thing. So this is the secondary product of the parietal or oxyntic cells. So if you see the main component, your main function of this HCl, so this HCl is going to create an acidic environment. What is the use of this acidic environment? This is the first important, first key player in the digestion of our food. So main digestion starts from the stomach, and there this HCl is playing a very significant role. So this creates an acidic environment. It also activates the pepsinogen into pepsin for the protein digestion. Then this a strong acidic environment, which is roughly 1.5 to 2.5 pH, that kills the ingested microorganisms which we normally ingest through our food, water, or different ingested foods. This also restricts bacterial growth in the stomach and prevent the intestinal infections. So this helps in many ways. If you see, it is known that the capacity of the stomach to secrete HCl is almost linearly related to the number of parietal cells. This is the normal basic structure of the intestinal stomach, where we are having different types of cells, and one of the important cell is this parietal cell, which we are going to talk today. So this parietal cell, so the amount of HCl production is normally linearly related to the parietal cells number. So now if more number of parietal cells will be there, more acidic production will be there. Less number of cells will be there, less production will be there. So this HCl is produced by the parietal cell, which is also called as augmentic cells. These cells normally have two faces. You can see here there are two surface. One is this side, and second one is the luminal side. So the side towards the gastric lumen is called as the luminal side. So this portion is called as the luminal side, and this outer portion is called as the basolateral side. So this side we are having the blood capillaries, blood vessels. Now, the three for the HCl production, there are three important ingredient is required. Uh, that is water, carbon dioxide, and chloride ion. How this is going to involve, that we will see through pictorial and through the different aspects how HCl is going to be produced. Whenever these parietal cells are going to be stimulated, it secretes HCl at a concentration of roughly 160 millimole, that is equivalent to pH 0.8. The acid is secreted into the large canaliculi deep invaginations of the plasma membranes, which are continuous or the lumen of the growth. So if we'll say like here, how this, so carbon, I have told you there are three important key players. First one is the carbon dioxide. So what this carbon dioxide, from where it comes? This carbon dioxide is basically hydrophobic in nature. So normally it cannot directly come into the cells. So it diffuses from the blood through the basolateral membrane. Basolateral membrane is the membrane which is toward the blood capillary blood vessels. So carbon dioxide diffuses from the blood through the basolateral membrane and reaches into the cytoplasm of the augmentic cells. Then second one is water, which is also coming from the blood through basolateral membrane only, but through a different channel that is called as aquaporine. Aquaporine is a channel which is normally responsible for the water transportation. We have different types of aquaporins. Means important. There are 7 important aquaporins we are having, and different cell will have different types of aquaporins. So here, this is aquaporine channel normally diffuses water from the blood through the basolateral membrane, and that water also reaches into the cytoplasm of augmentic cells. Inside the augmentic cells, because we are having carbon dioxide also, we are having water also. So this carbon dioxide and water combines together in the presence of an important acid that is called carbonic anhydrase, gives us carbonic acid, that is H2CO3. And this H2SO3, that is a carbonic acid which normally dissociates into H plus and bicarbonate ion HCO3 minus. Now, so inside the parietal cells, we started from carbon dioxide and water, and we are having H plus and bicarbonate ion. So now if you'll see, this H plus ion which we have got from the bicarbonic means carbonic acid pumps into the luminal side, lumen, through the proton potassium H plus potassium ATPase channel, which is present on the apical membrane or the luminal membrane, you can call it. Now this H plus, which is coming into the lumen, decreases the pH of the gastric lumen and makes environment very strong acidic. And the remaining bicarbonate ion that is going to be transported out of the basolateral membrane into the blood circulation in exchange of chloride. So chloride comes inside the parietal cells and bicarbonate ion going out of the parietal cells into the blood circulation. So there is an exchange between the chloride and the bicarbonate ion. Similar things happens in our RBC also, and that we are going to call chloride shift. So the outflow of bicarbonate into the blood results in a slight elevation into the blood pH, means blood becomes more alkaline because more bicarbonate is reaching to the blood circulation, and this is called as alkaline tide. This process serves to maintain the intracellular pH of parietal cells, because if this bicarbonate ion will not go outside the parietal cell, so parietal cells cytoplasm becomes more alkaline, and that is not required or not that is going to harm the cells. This is the process how it is going to happen. So we are having H2O and carbon dioxide coming through the basolateral membrane into the cells. There we are having carbonic anhydrase. This carbonic anhydrase combines this H2 and CO2, gives us H2CO3. This is going to dissociate into H2CO3 and bicarbonate and H plus. This H plus moving out in exchange of potassium through a proton potassium ATPase channel. So H plus goes out into the lumen and potassium comes inside. This potassium normally moves into the cyclic manner. So this H plus will be there. And then this bicarbonate ion is going into the blood circulation through a shift proton, that is a in exchange of chloride. So chloride ion comes into the lumen. From lumen it is going to the through the cells, it is coming into the lumen. So in the lumen we are having H plus, we are having Cl. So this is going to combine together and gives us HCl. So this is the overall process of acidic productions. What you are going to see here, there is the acetylcholine, histamine and gastrin. These are the three main regulator of the acid productions that we are going to talk about. So regulation of acid productions occurs through three important regulators. What are the regulators? So parietal cells bears the regulator of uh, these regulators normally activates receptors, and once the regulator receptor will be activated, they increases the activity of proton potassium ATPase channel. So ultimately more acidic production will be there, and these three regulators normally regulates through neuronal, paracrine and endocrine controls. What are those regulators? That is acetylcholine, gastrin, which is coming from the G cells, and then histamine, which is working through the H2 type receptors, and histamine is normally produced from the enterochromaffin like cells and the mast cells. So histamines from the enterochromaffin cells may well be the primary modulator, but the magnitude of the stimulus appears to result from a complex additive or multiplicative interaction of all the three molecules, all the three regulators. So like such as, if you see the examples, the low amount of histamines, whenever histamines concentration will be very low, it normally which is normal generally coming from the mast cells in the normal cells. So it comes into the gastric mucosa and weakly or the poorly stimulates for acidic production. So only a small amount of HCl will be produced. Similarly, if you are having a small amount of acetylcholine, only a small amount of HCl will be produced. Similarly for gastrin also. So a small amount of gastrins, a small amount of HCl will be produced. But when all three low level of all three will be there, they normally strongly activates the HCl production. So even a small amount of acetylcholine, gastrin and histamines together activates HCl production in very huge amount, very high amount. Second function of this histamine is histamine effects on the parietal cells to activate adenylate cyclase. This adenylate cyclase normally leading to the elevation in the intracellular production of cyclic AMP concentration, and this cyclic AMP is responsible for the activation of protein kinase A. Protein kinase involves in many different receptor regulators of different many signaling cascades. So due to histamines, cyclic AMP production will be there because histamine activates adenylate cyclase. So more cyclic AMP, more protein kinase A activated. Once this protein kinase A is going to be activated, it is phosphorylation of the cytoskeleton protein involves, and that helps in the transportation of this channel. So normally this channel lies into the cytoplasm. So this is a cell. So normally this channel lies into the cytoplasm. So because of this histamines and the cyclic amine protein kinase A, this channel moves from here to the plasma membrane so that exchange will take place. So binding to the acetylcholine and gastrin both results into the elevation of intracellular calcium concentration, and that calcium also helps in the transportation of this sodium potassium proton and potassium channel. Other than these three regulators, these three are the main regulators, but there are several other additional mediators also helps in the gastric acid secretions, which infuse into the animal and people in like such as you can see calcium, because calcium you have seen how calcium helps in the movement of this channel into from cytoplasm to the plasma membrane. Then we are having bombesin, both stimulates gastric acid gastrin releasing and opiate receptor have been identified on the parietal cells. So there are a number of regulators are there. A variety of substances are capable of reducing gastric acid secretions also. They are also regulators, but they decrease the productions, such as prostaglandin E2, number of other peptides, you can call it like secretin, gastric inhibitory peptides, glucagon, somatostatin. These normally reduces like prostaglandin E2, secretin, and somatostatin may be the physiological regulator. Somatostatin inhibits the secretion of gastrin and histamine and appears to have a direct inhibitory effect on the parietal cells. So this is how HCl is going to be produced, and HCl is going to be regulated through three important. So these three normally activates the productions, increases the productions, and some of the uh, inhibitory production means who regulates in the negative manner, that is somatostatin, prostaglandin E2, secretin, gastrin inhibitory peptides, glucagon. So this is all about. Hope you have understand it. Next day we will have another video. So thank you very much. If you have not subscribed my channel, you can subscribe it. If you like the video, press the like button. If you have any query, any comments, any suggestions, please write into the comment box. Have a nice day.